Animal Development, Chapter 53. Animal development is not how the cute and cuddly grow, but the process of how the cute and cuddly get here. Animal development has 4 stages: fertilization, cleavage and blastula, gastralization, and organogenesis. Fertilization is a fusion of a sperm and an egg, or, as the textbook puts it, male and female gametes. Fertilization is usually external for aquatic animals and internal for terrestrial animals. There are 3 events that make up fertilization: sperm penetration and membrane fusion, egg activation, and the fusion of the nuclei. Sperm penetration and membrane fusion. In order for membrane fusion to occur, a sperm must penetrate the protective coats of the egg to reach the plasma membrane. The fusion of the membrane and sperm is what starts embryonic development. Egg activation. When an egg is activated, it builds a defense to prevent polyspermy, when multiple sperm fertilizes one egg. If more than one sperm were to fuse with the egg, that would lead to polyploidy, having more than 2 sets of chromosomes, which is incompatible with animals but frequently found in plants. Fusion of the nuclei. The haploid sperm and egg nucleus fuse and form the diploid nucleus of the zygote. However, the haploid nucleuses do not fuse in mammals. Instead, the 2 nucleuses break down and a new nuclear membrane forms around the chromosomes. The cleavage and blastula stage. Cleavage is the rapid division of the zygote and does not increase the overall size of the zygote. A blastomere is an individual unspecialized cell in a tightly packed mass of cells. Blastomeres are formed during and by cleavage. A blastula is an early embryo that will typically have a hollow center filled with fluid. A zygote is like the Earth. They both have 2 poles, but an egg's poles are called animal and vegetal. Cells that form the animal pole will later go on to forming the external tissues, and the cells of the vegetal pole form the internal tissues. The 3rd step in animal development is gastralization. Gastralization forms of 3 primary germ layers: endoderm, mesoderm, and ectoderm. The endoderm. The endoderm is the innermost of the 3 primary germ layers of the embryo. It forms the digestive tract and its derivatives. The mesoderm. The mesoderm is the middle of the 3 layers and forms bodily tissues and structures such as the tissues, bones, and muscles. The ectoderm. The ectoderm is the outermost layer. It forms various tissues and structures such as the nervous system, eyes, and ears. Organogenesis is the 4th and final step in animal development. Now that the basic body plan has been laid out thanks to gastralization, the stage is set for organogenesis to occur. Organogenesis has to follow closely behind the end, if not before the end, of gastralization, because organogenesis occurs by interacting with cells of the germ layers to develop the bodily organs. Simply put, organogenesis is the development of the organs.